Okay, so yeah, maybe it's a bit unconventional for us to be two on stage, but we are a super large team, as you will see in the acknowledgement section. So we're only two here, but it's really the work over many years of dozens of people. So yeah, thank you for having us here. And Olivier and I, we are so pleased to present the work of the International Brain Laboratory that aims at mapping the relationships between the extracellular electrophysiology signals, the anatomy, and the gene expression patterns in the mouse. So we'd like to begin with this sort of vanilla statement that our brain is an organ with a very particular anatomical structure. So if you take first the piece of cortex on the left here, we know that it's organized neatly into layers. We have different cell types, as Kenneth just showed before. And so there is a very well-defined anatomical structure. But if you were to take another brain region, so take cerebellum here on the right, and the structure is completely different. So there is, it is organized, but it's completely different. There are other kinds of cells, Golgi cells, and so on, and the innervation patterns are different. The long-range projection patterns are different. So the question we are interested in is whether this anatomical structure is at the origin of function. And here, what we mean by function is really basic electrophysiological signatures. One example is the sharp wave recall in hippocampus, and we'll go into details of really what we call signatures. So it has nothing to do with complex behavior. It's really basic electrophysiological properties. And we want to study this link between the anatomy and what we call these electrophysiology signatures at the whole brain-wide scale. So the first step that we are going to do here is look for atlases of the anatomy at the whole brain scale. So here we really want to produce the Allen Institute. There are other atlases out there, but for the sake of this particular talk, we're going to use 3 atlases from them. The first one is the brain region parcellation atlas. So for a given region in the brain, you get the label of the region it's in, like CA1 for hippocampus, for example. The second kind of atlas we're going to use is the gene expression atlas, but at the voxel level. So for a piece of tissue, basically, a set of genes are looked at, and we know whether they're expressed or not. Sorry, that's too small. And the third kind of atlas we're going to use is their more recent cellular atlas, whereby cells are located in the brain, and for these cells, you have a set of genes that are expressed. So what we hope to throw in the mix is what we call the electrophysiology atlas. So it would be a whole brain atlas of basic electrophysiological feature, and now we're going to show you how we construct it. So we are using a very large data set composed of more than 1,000 insertions done in the mouse brain. So it's used, we use neuropixels probe in different mice, in more than 200 mice. And if you were to compare this with our recently released brain-wide data set, it's nearly the double in terms of the number of insertions. And the reason why that is that for this particular analysis, we don't care as tightly for the particular behavioral performance of the animal. So it's a data set that is very large in size. You can see like it's over 50 terabytes. And now we're going to process it and extract some signals from it so that then we can do modeling and so on and fit this into memory. So the kinds of signals that we extract are the following. So if you're not familiar with electrophysiology, just to show you very quickly what the raw data looks like, it's this gray voltage trace. This is a very short time window that we're looking at, around 15 milliseconds for each of the electrode channels here. So for a given electrode, we span different brain regions. This is like hindbrain and midbrain here. And for a given electrode, we can extract different signals at different levels. So the first is on the action potential signal. For example, we can take the root mean square of that signal. That's like a proxy for the spike amplitude or the spike rate. We also take features at the LFP level, such as the power in the different bands, like theta bands and so on. And we also extract features at the spike waveform level, like the polarity of the spike. So this is what we extract from this data. So it's much smaller in the end than the actual raw data. But before going into the analysis of such signals, we're just going to show you so that you have a feel different kinds of regions. So this is the raw data for 3 different recordings that span different regions. So again, hindbrain and midbrain for the first one. The next is cerebellum, yellow, then it's cortex, hippocampus, and thalamus. And you can see, hopefully by eye, that they are very different. So we basically want to extract signals that mark these differences. And it's a very short period of time here. So that's why we are hoping that hopefully these are intrinsic electrical properties that don't have so much to do with behavior, but we can keep that question for later because we know that it's important. So these are the signals we extract, and what we care now about is to see whether there are differences between brain regions and what are the features, electrical signatures, if you like, that would differentiate these different brain regions. So throughout this talk, we are going to use, for you to have a feel, a very coarse parcellation of the Allen Atlas. We call it the Cosmos parcellation. So for example, all of the cortical layers, all of the cortical regions are lumped into one category called isocortex. So if you take these categories and you draw now distributions here for one particular feature, that is the peak to trough duration of the waveform, you can see that different brain regions have different properties, basically. So for example, here, if you take cerebellum in yellow or the midbrain and hindbrain, they have very small peak to trough duration. In other terms, the spikes are really narrow compared to the other regions. To give you a feel, these are like two randomly selected units, one from the midbrain, one from the isocortex, and you can see hopefully by eye that the one in the midbrain is indeed much narrower than the other. So we can do this for all of the different properties that we look at. So here first it's the LFP, so the PSD, the power in theta band. The other is the RMS, so the amplitude, if you like, of the actual potential band. And you can start to see that different properties will distinguish different brain regions. So another way to look at this is spatially. So before we were looking at distributions, but now if we plot this onto an atlas, here we're making a website to navigate this. You can see, for example, that the AP-RMS is very strong, mostly in the thalamic region. And if you take another feature, here this is the LFP-RMS, it's basically reversed. So here it's mostly located in the hippocampus, in the cortex. And if you switch back and forth, you could see that they are almost anti-correlated. So this is what we've constructed. And now the question is, what are the use cases for such an electrophysiology atlas? We are going to go through 3 of them. I will present the first one, and then we'll switch to Olivier to present the 2 next ones. For the first one, we are not so much going to go into the gene expression atlases. We're mostly going to look at the correspondence between the electrophysiology features and the mouses here. Okay, so the first thing is to say the assignment between the brain region labels. So this is what we see here on the far right, these Allen colors, and the actual electrode channels that you have is not trivial. So we're going to go through one simple example that's with you. So first, if you were to mark by eye the boundary between the hippocampus that you see on the green here, I'm sorry, and the thalamus that is pink, the question is, where would you mark this line? So if you're a human and you're well versed in the literature, you know that the LFP power in the hippocampus should be really high. So that's kind of the yellow blob that you see on the left. And you know that the thalamic region often have a lot of spikes, so the RMS-AP should be rather high. So you can be confident from the literature that this line should be marked around the depth 2000. So that's kind of easy. But if now I were to ask you, please mark the boundary between NP and PO, these like deep thalamic nucleus that nobody knows nothing about, you wouldn't know where to put it. Okay, it's kind of hard from just this plot. So what we provide is a way to basically rank the different features first, so that you know which ones are the most informative. And then if you replot just here the PSD theta for this different region, you can see, okay, the NP region actually has a much higher value than the PO, and we can help you to draw this line. So obviously, we just did this exercise kind of by hand, but the whole point is to make an algorithm that does this for you. So this is what we are trying to build. So it's called a brain region decoder. Here, we're just going to present you the work that uses individual electrode channels at once, but we have work in the pipeline so that we use the whole probe geometry and so on. So if you have questions, please feel free. This is preliminary work. So for a given electrode channel, we take all of the features that we extract and we do gradient boosting. So it's a decision tree, and we get a brain region label as output. And to give you an example of what it looks like, On the left, so if you take the second column, this is the real region arcs, the ones marked by a physiologist who knows where these boundaries should be. And on the far left, it's the prediction of the algorithm. So each of these lines are a single channel. So it's, again, at the cosmos level, so this third coarse parcellation. And what the algorithm gives you actually is a cumulative probability of which brain region it should be, and we just take the highest as a label. And to give you a feel of how good this algorithm performed, this is the confusion matrix. It's pretty good overall for this coarse cosmos parcellation. There are some regions for which it's not good. We know why. We think the parcellation from the LN has some tweaks to be made to it. I'm happy to take questions at the end as well for this. So that's what we are trying to build, is a brain region decoder. And finally, to give you a feel of why it's useful, let's take the classic case of an experimenter wanting to put their probe in the brain. So that would be the planned trajectory here in black, and say, I want to go just so deep so that my electrode passes the hippocampus in green. So the cosmos line here would be what regions they think they would be in. So they think, okay, great, I should stop here because I've actually hit my target. But what the decoder gives you is that you're actually not there at all. You're still very much in the hippocampus. So we think of it as a tool to minimize targeting error. So that's the first application. And with that, I will pass it to Olivier for the next two. Thank you.
To relate the anatomy with the electrophysiological features, and now we're going to look at how those electrophysiological features relate to gene expression. And there are two good reasons to do it. One of them is naturalistic. We would like to understand a bit the biological concepts that are behind those electrophysiological features. Another reason is a bit more practical, is the fact that we may use the spatial structure of the gene expression to interpolate our electrophysiological features on the full 3D volume. I'm going to show a bit why this is important. On the left, even if we have 1,000 experiments with superhuman experimenters, if you slice through the brain and you look at the number of neurons, which is a proxy for the number of channels, you can see that it's still quite sparse at the 200 microns parcellation. And you can see on the coronal slice the same thing at the bottom. So the idea is that if we get the gene expression patterns in 3D, can we use this in order to interpolate our data samples on the full atlas? So this is an example on the right of the results of such compared to the parcellation average. So the parcellation average is over 308 regions. We're going to take the average of the electrophysiological features, and this is going to be the voxel value. And on the right, this is the interpolated values. So in practice, what does it mean? We have 4,000 genes on the LN gene expression atlas. It means that we have more than 4,000 volumes, full 3D volumes of gene expression that are sampled at 200 microns. We also have the LN brain cell atlas, and this contains around 9 million cells of clustered cell types. And in this case, we can aggregate those cell types, bin those cell types at the same resolution as the LN gene expression atlas. From there, for each voxel, we have a vector that is around 4,000 genes that we call the local gene expression profile. And we are going to encode, we're going to use this local gene expression profile to predict these features. In practice, this is a bit too much, so we're going to use a PCA, dimensionality reduction, in order to use something like 275 genes in order to predict these features. This is an example of the predictor. On the left, we can see the real PSD data. So this is the recorded data, the feature, which is the PSD of the delta bound of the LFP. On the right, this is the model prediction. Here, our insertion, it's upside down. It goes through the visual cortex, somatosensory cortex on the bottom, through the hippocampus, and then the thalamus on the top. So we can do one model, and we can repeat this, iterate for all of our features. And this is going to be the result. On the left, this is one test probe, and this is the model prediction on the right. We can see the same anti-correlation pattern that we've seen before between the LF feature and the AP feature. Here, the RMS AP is anti-correlated to the PSD of the alpha. So that's for one insertion, and we're going to go further and look at the performance of this model through the full cross-validation set. So on the left, this is our null model. This is the baseline model, which is the parcellation average. So for each region, we take the training set samples, average them, and the test sample, we take the difference with the test sample within the same region. This is the R square explained for this model, and on the right, this is the gene expression model R square. We can see that the gene expression model is much better at predicting these values in 3D space. One of the reasons behind this may be because the parcellation is too coarse, and maybe within one region there is some differences in the features. So let's have a thorough look at this for one region. So the CP, Gaelle, for example, which is a large region. And here we can see on the right that the gene expression for this region is quite unhomogeneous, especially for CRLF2. So we can hope that the gene expression pattern is going to give us better resolution. Another thing I would like to point out on this screen is that you can see the gene expression patterns, how tightly linked to the spatial structure of the brain. I think this is amazing when we look at the genes like this. Yeah, gene expression. So here is the example for the cardo-putamen coronal slice, and we can see that the parcellation average on the left, which gives us a single value, on the reconstructed atlas, we get some more specific, more inhomogeneous values. Here I must point out that we're still working on the fact that those little areas, they are part of the parcellation problems that we identified and we are after. So happy to answer questions about this. The next part is what such a model could be useful for is to look a bit at what gene is involved, is correlated with some electrophysiological feature. So here we are going to rerun the model, each time taking out one gene, and to see how much variance explained is going to be lost by removing this gene. And here we have the ribin protein example that we saw at previous talk again. And this is correlated with the RMS of the low frequency band. So here it may be just because those things are both expressed in the hippocampus, but there may be some biological processes underlying this. So there is some future work where we need to continue on this. First is to create a combined cell type and AGR in this picture predictor. I've shown the result only for the AGR, so which is the gene expression, overall gene expression. Here is the result for only the cell type atlas for the ALN. I think we can combine both to see if we get a better model. We like to add new features as well, new electrophysiological features, extract them from our data. And we want to add some controls for small nuclei. What I mean by that is that we need to look at those little areas where we have a big mismatch between the gene prediction and the ALN parcellation with anatomy. The last example I'm going to talk about is the cell type classification example. It's much shorter, but it's actually much more classical. It's something that's been done before. So we are going to take 35,000 clusters throughout all the brain and do waveform classification on those. We take the templates, single trace templates for only the good units, and we're going to run them through WaveMap, which is a classifier, and classify those waveforms. So here, what we have is that we have 10 waveform clusters, and they are not cell type, they are waveform clusters. And the idea is to say, since we have recordings throughout the brain, how do they going to map on the 3D structure of the brain? And this is the first results that we got out of this, and we can see that the clustered waveforms have a spatial organization, have a very spatial organization. So here what is important is that we see the cortex cells that are the most studied are very different from what we find in the middle here, and again very different from the midbrain, the hindbrain, and the cerebellum. And the value of this work would be to try to work at a whole brain scale. So here there is a lot of work still to be done for this part. First is to refine the spike sorted features. We want to compute correlograms, probably time scales, and especially use multi-trace templates. I think that's quite important given the structural organization of the brain. Then we want to use spike population models. Here, if we look before spike sorting, we can say, look at all of the spikes that are found in a channel, and to see the distribution of the spike features through this channel could inform us about where we are in the brain. And the next part will be how to relate those spike populations to the cluster populations that are found in the alien brain. So in conclusion, we've shown 3 applications for this. The first is the relationship between anatomy and electrophysiology can help us build some tools, online tools, in order to predict where the probe is inserted in the brain from a single channel. Another thing is that the relationship between the genomics and the electrophysiology can help us to create electrophysiology features volumes. And this could be useful as description of the brain. That could be used as a baseline. So for example, if we do some kind of experiment on a mouse model, we can see how they differ from our baseline atlas. At last, it could be a useful resource for pre-processing, such as spike sorting. So typically here, what I mean is that extracting the features from a single channel could help us define specific parameters for this channel when we do spike sorting, because we know that the thresholding may be different whether we work in cerebellum rather than we work in cortex or thalamus. And at last, one big takeaway of this work, at least for us from the engineering perspective, is that working on feature engineering at scale is not trivial. And this is something that we will have to get better at if we want to work at brain-wide models. Here we are talking of thousands of insertions, terabytes and terabytes of data. So each time we want to recompute a feature, this is not something that can be done lightly, and engineering matters. And with this, I would like to thank especially the neurophysiologists that collected the data. This is a huge amount of work, and those are the persons that contributed to this brain-wide map. And at last, I want to thank the team working on this task force, the idea as a general, and of course our founders for their trust in our ideas. And thank you.